So this is the model for the DNA, and you know that this model was given by Watson and Crick, and for this they were awarded Nobel also. So according to them, DNA consists of two strands. DNA consists of two strands, as you can see that, and these strands are highly coiled. As you can see, they are just coiling around, forming a spiral staircase. So we say that it consists of two polynucleotide strands. It consists of two polynucleotide strands highly coiled with each other, forming a spiral staircase, or we can say forming a double helix structure. So in this you can see that as I told you that DNA is formed of like the monomer which forms the DNA is a nucleotide which consists of three parts. One is the phosphate group, other is the pentose sugar, and third is the nitrogenous base, and this you can easily see in this figure. So if we talk about the strands first, so you will see that I have mentioned one strand as 5' and its end as 3', and the other strand as 3' and its end as 5'. So actually these two strands are anti-parallel to each other, and that is indicated by this 5' and 3' and 3' and 5'. So they are anti-parallel to each other, right? And now if we talk about that like what forms its backbone, so we have seen that backbone is formed of phosphate sugar group. So as you can see that phosphate linked with sugar, linked with phosphate, again with sugar, this goes on repeating, and this is forming a strand actually. So the backbone of the strand is formed by phosphate sugar groups, as you can see them, and you need to remember that they are coiled, they are just coiling around, and it is a double helical structure and forming a spiral staircase like form. And in this we can see that the two strands are linked to each other. The two strands are actually linked to each other, and how they are linked? They are linked by the bonding between the base pairs, that is the nitrogenous bases. So what happened here is, like if purine appears at this place, so at the similar place on the next strand there is a pyrimidine, and they are linked by the specific hydrogen bonds. As we know, adenine bond with always thiamine with double bond, thymine always bond with adenine with again double bond, cytosine bond with guanine with triple bond, and guanine also bond triply with the cytosine. And this is how the base pairing is occurring, and these bonds are the hydrogen bonds. So that means you need to remember that the two strands are anti-parallel, and secondly the two strands are base pairs are complementary to each other. That means if adenine appears on this side, on the similar side there is a thymine, because we know that adenine always pair up with thymine and the bond between them is double bond. You need to remember that. And if guanine appear on this side, on the next side we will have a cytosine, and they will be again paired with the triple bond, because we know that the pairing between the cytosine and the guanine or the cytosine is always a triple bond. So this is how the base pairing occurs, and they are just linked with the weak hydrogen bonds. Now after the occurrence of 10 base pairs, there occur a helical turn. This you can see, this is a turn, this is a turn. So after each 10 base pairs we have a turn, and this turn can be left sided or can be right sided. It is not a hard fire rule that we will get a left handed turn or we will get a right handed turn, but definitely there will be a turn after each 10 base pairs. So this is what is happening. So if we talk about the height of the complete DNA, we will see that the complete height is 3.4 nanometer. The complete height for a DNA strand is 3.4 nanometer, and if we talk about the width, this width, it is approximately 2 nanometer. So you need to remember this. So this is what about Watson and Crick model. So like if you get a question like this, that to explain Watson and Crick model, you are going to explain not directly the structure, you need to explain about the nucleic acid, nucleotide, a basic introduction about the nucleotide, and then come on to the structure. So I will just repeat an answer for you, just listen to me carefully. You are going to write that DNA is a polynucleotide formed of a monomer nucleotide, and each nucleotide consists of three things, that is a phosphate group, pentose sugar and nitrogenous base. Then explain the sugar, as I have told you that DNA consists of deoxyribose sugar, and you also know what is the formula we discussed in the previous lecture. So that you have to draw, and then with that you need to mention that what are the nitrogenous bases. We will classify nitrogenous bases into purines and pyrimidines, name them adenine, guanine and thymine and cytosine. cytosine, and then also mention that what kind of pairing take place between them, that adenine bond to thymine with double bond, guanine bond with cytosine with triple bond. After doing that, then come on to the model, then write that this model is given by Watson and Crick, and they are awarded nobly for this this work. And according to them, DNA consists of 2 polynucleotide strands. DNA consists of 2 polynucleotide strands which are highly coiled, forming a double helix structure. We can also compare this structure to a spiral staircase. And what we will see here, that the backbone is made up of phosphate and sugar group, and 2 strands are antiparallel and complementary. Do not miss these words, complementary and antiparallel, these are most important. And you can see that 2 strands are connected to each other by weak hydrogen bonds between purines and pyrimidines. By saying that these strands are complementary, we mean that if there appear adenine, so on the same side we have the same pyrimidine which links to the adenine, that is the thiamine. And after 10 base pairs, there occur a helical turn, and this turn can be left-handed or a right-handed. And likewise, we will be mentioning the height also. So the height for the complete DNA strand is the 3.4 nanometer, and width is 2 nanometer, and after 10 base pairs, as I told you, there is a helical turn. So this is what about the DNA helical structure given by Watson and Crick, and now we will study that how it helps in the biosynthesis of proteins, or how it differs from the RNA, and what kind of function it is actually performing. And one more thing you need to remember, that we can actually separate these strands. We can actually separate these strands, that like 2 strands. How? By heating. Like if we heat them at appropriate temperature, these 2 strands get separated, and this is something called as melting. This is something called as melting, and it occurs definitely at specific temperature, that is called as melting point. And if you cool them, these two strands can again coil with each other, and that process is something called as annealing. So melting is separating 2 strands which occur on heating. Annealing is joining again, making them again coiled with each other, and how it occurs? By cooling. And so there is a specific temperature at which melting and the annealing take place. So we will see that, because melting and annealing are playing an important role in the biosynthesis of proteins and transfer of hereditary characters. We will discuss that. So this was the Watson-Crick